All right, so we're going to spend our time this evening going through some of the, I think, maybe the more important aspects of lipids to discuss some of the features of them and such like that, and to point out some of the reactions and different things. How you doing, Kim?
Good, how are you?
Good. Before I get going, did you have any specific questions, or
Yes, I did.
I want to take care of that first and foremost.
So on page 1 or 521,
Okay.
question 1520. So I kind of struggled with 1519.
Okay.
So I wanted to kind of get help with 1520
Sure.
to kind of help with 1519.
Okay. No problem. So 15, 19, 20. Okay. Okay. So, all right. So 15, 20. Let's get the pen. All right. So draw the condensed structure. Draw the condensed structural formula for the ester in jojoba wax that is formed from arachidonic acid, a 20 carbon saturated fatty acid, and 1 docosahexaenol. All right. So essentially, the way that most lipids are made is by esterification. So if you remember back to the discussion of esters, if you have a carboxylic acid plus an alcohol, we can combine them together by taking the OH group off of the carboxylic acid and the hydrogen of the alcohol and bonding the C double bond O carbon of the carboxylic acid to the alcohol oxygen. And when we put that all together, that gives us our ester functional group. So this particular process that we're seeing here in 1520 to essentially form a wax is going to be something that's very similar. Now, in terms of the fatty acid, so they tell us a 20 carbon saturated fatty acid. So what do we know about fatty acids? We know that at one end we have a carboxyl group, and then basically we have a carbon chain, which could have all carbon to carbon single bonds, which you find in your saturated fatty acids, or they could have 1 carbon to carbon double bond, which are your monounsaturated fatty acids. And then you could have more than 1 carbon to carbon double bond, which are your polyunsaturated fatty acids. So in terms of a 20 carbon saturated fatty acid, at one end, we're going to have the carboxyl group. So that would essentially, in my mind, sort of be carbon number 1. And then basically everything else that's there is carbon to carbon single bonds. Now, one of the shortcut methods of writing the structures of fatty acids is that when you have CH2 groups that are connected to each other over and over and over again, is to put that in parentheses with the number of times that it repeats as a subscript on the outside. So in this case, if we want 20 carbons total, you would have 18 consecutive CH2s. And then after that last one, the 18th and final, you would have CH3. The other way that you could sort of draw this would be to sort of put the carbon chain in skeletal form. So you'd have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, and then your 19th and final carbon. So those are a couple different ways. You could draw all 18 CH2s out, but that's going to make it a little bit difficult. Now for the alcohol, so now we have the fatty acid. They give us the structure of the alcohol. So the alcohol is a 22 carbon alcohol. Because you have CH3, then you've got 21 consecutive CH2s, and then you have the OH. So how do we draw the product? From the carboxylic acid, or from the fatty acid in this case, you take off the OH. From the hydrogen of the alcohol, you take off the H. So the byproduct of the reaction is going to be water. And then you form a bond from the C double bond O carbon of the fatty acid to the alcohol oxygen. So in this case, the structure of the wax that we're going to get, and again, I like to draw the fatty acid portion first, CH3, CH218, C double bond O. Then you have your connection to the alcohol portion. So there's the connectivity between them right here. And then you'd have 21 consecutive CH2s. And then after that, you'd have CH3. So essentially, this is the structure of the wax that would result in this example. Now, I think actually they might even give this structure or talk about this structure in the book. And I think they actually give a different structure. I think instead of 21, they actually put 19. Because when I've graded homeworks from this chapter in the past, students have drawn the 19 and they point out that it's either in that way in the book, or if they look it up, they give that structure as well. So I usually just, in this case, sort of just tell them we got to go by what the book has, and usually it's not a big issue. But so this is the structure that, based on the information given, we would come up in this example.
Okay, so that one is correct for that number. What number are we on? 1520.
Yes.
Okay, because they gave us that little, that is the alcohol that they gave us.
Correct.
Okay. Yeah, that's where I got a little confused.
Okay.
So is that structure docosano? One docosano?
I believe so. I don't
Okay.
really come across that one too often. But
Okay.
but yeah, I believe that is the structure of that alcohol. Unless it has unless it's got like CH219 instead, because as I said, I've seen different variations of answers submitted. But
Okay.
either way, to draw whether it's a 21 or 19, the way that we would draw the product would be exactly the same. And that's sort of the key idea.
Okay. So I think I just have one more.
Sure.
1525 of that same page.
Okay. So safflower oil is polyunsaturated, whereas olive oil is monounsaturated. Why would safflower oil have a lower melting point than olive oil? Okay.
So I understand the difference between saturated and unsaturated. And I saw the back on the book, it talks about how the double bonds, but I just don't understand why. What is about the double bonds to make it have low melting point?
Sure.
Take less energy.
Okay. It's not a problem. So the main forces of attraction between fatty acid molecules are dispersion forces between the carbon chains. So we saw dispersion forces earlier on in the semester, where you have a momentary shift in electron density in something that's nonpolar, which causes a response in something else that's close to it. And then all of a sudden, essentially, these two things are attracted to each other. So you have two nonpolar things that are automatically which you wouldn't expect to be attracted to each other, now actually attracted to each other. Even though fatty acids have carboxyl groups, where you would think hydrogen bonding would be really in play, because of the length of the carbon chains, the main attraction between fatty acid molecules are dispersion forces between the carbon chains. And to melt the fatty acid, we have to break the dispersion forces present. How do we do that? We supply heat. Heat, of course, is the form of energy. Now, the dispersion forces are really impactful if the carbon chains of the fatty acid molecules fit together in a regular pattern. And that really is what you see in saturated fatty acids. You know, if you go back and look at the textbook, they show you the picture where, you know, you have the saturated fatty acid molecules, and they show you like the Pringles potato chips, where they stack like one on top of the other, on top of the other, on top of the other, on top of the other. Because the saturated fatty acid molecules are able to fit together in that regular pattern, dispersion forces are really able to be present between the carbon chains. So what that means is that at room temperature, because room temperature is a measure of the heat or energy that is present, at room temperature, there's not enough energy to break enough dispersion forces so that a saturated fatty acid would go into the liquid state. And therefore, essentially, your saturated fatty acids are solids at room temperatures. Now, your unsaturated fatty acids have cis double bonds. You know, the textbook talks about the fact that, which is true, that because each of the double bond carbons have one hydrogen, you could have cis and trans isomerism. But in most naturally occurring fatty acids, the type of what you have around the carbon-to-carbon double bonds is you have a cis configuration. What this does is that it causes let's see what's going on. So it causes what's going on? So this causes the carbon chain to bend, which gives
Okay, Mom, I got it now.
each molecule an irregular shape. So if you look at essentially a saturated fatty acid molecule, you know, the carbons don't go in a row, but if you sort of follow the pattern, you know, essentially, if you start, you could sort of trace a line straight across to the other end. However, for an unsaturated fatty acid, once you get to that double bond, essentially what it does, and I probably didn't draw it as well as I probably should have. So in terms of what an unsaturated fatty acid looks like, for an unsaturated fatty acid, if you start relative to the carbon-to-carbon double bond, you have something that more looks like this. Because the molecules have an irregular shape, your unsaturated fatty acid molecules do not fit together in a regular pattern. And therefore, there are minimal dispersion forces present between the carbon chains. And therefore, in order to go from a solid to a liquid, it takes much more energy much less energy to break the dispersion forces between unsaturated fatty acid molecules as compared to saturated fatty acid molecules. And therefore, for the most part, at room temperature, there's enough energy that's present to usually melt that unsaturated fatty acid. And therefore, for the most part, your unsaturated fatty acids are liquids at room temperature. And that's essentially why they have a lower melting point. Your saturated fatty acid molecules are able to fit together in a regular pattern, which allows dispersion forces to occur between the carbon chains. And your unsaturated fatty acid molecules, because of the cis nature of the double bonds, essentially the carbon chain bends at each cis double bond, and that gives the molecules an irregular shape, and therefore dispersion forces are not as readily able to exist. Now, the more double bonds that are present, the more the carbon chain bends, and the more irregular the shapes of the molecules become. So as you go from, let's say, a monounsaturated fatty acid to a polyunsaturated fatty acid, the polyunsaturated fatty acid molecules are going to have more and more of an irregular shape, because every time you have a cis double bond, the carbon chain is going to bend. And therefore, the more and more irregular the shapes of the molecules become, the lesser the number of dispersion forces that are going to be present. And that's why in the example 25, when they talk about sunflower oil being polyunsaturated versus olive oil, the sunflower oil would have a lower melting point because it's got more carbon to carbon double bonds, and therefore their molecules have more of an irregular shape, and therefore there are less dispersion forces between the molecules of sunflower oil, safflower oil, as compared to olive oil. So it takes less energy to break the dispersion forces in safflower oil, and therefore by the time it melts, the temperature is lower. Okay, good, thank you. All right, any other, all right, so I think that was, I'll just pull up the chat feature. Okay, no problem, thank you. All right, so then let me go back to some of the stuff I was going to talk about, but I'm glad there's questions. All right. So let's see, let's get this back. So got some of the introductory stuff about lipids. You know, we just talked about fatty acids. So let's go on and maybe talk about some other stuff that could be important. Yeah, let's talk a little bit about triacylglycerols. All right. So essentially, the main type of lipid that sort of comes into play in terms of in the body, and not to say that's the only type, but one of the main types that you talk about are where essentially your fatty acids are stored. And your fatty acids are stored as part of essentially fats and oils, which are collectively known as triacylglycerols. And essentially the way that triacylglycerols are made is they're made by essentially three sort of esterification reactions using the same alcohol. The alcohol component of a triacylglycerol is always going to be the same. The alcohol component is always going to be glycerol. Glycerol is a trihydroxy alcohol that has the following structure. When I look at a triacylglycerol, I like to look at it as the backbone of a triacylglycerol structure. The difference between triacylglycerols is the fatty acids. The three fatty acids that can be used to make the triacylglycerol can be the same, or they can be different. So a lot of times in the triacylglycerols that you come across, they are essentially mixed triacyl glycerols that have different fatty acids that are present. And it could be saturated, unsaturated, it could all be saturated, it could all be unsaturated, and such like that. And again, it's really esterification that's used to make the triacylglycerols. You take off the OH groups from the fatty acids, and then you take off each of the OH hydrogens from the glycerol. And when you combine them together, that gives you your triacyl glycerol. So here's an example of a structure of a triacylglycerol. In this case, it's actually made from the same fatty acid three times. 1, 2, and I'll just 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Yeah, so it's basically made 3 times using the same saturated fatty acid. If you take a look, right, I like to call the glycerol the backbone of the structure. And if you take a look, you've got C double bond O connected to oxygen, connected to carbon. And when you have that functionality associated with it, what do we have there? Of course, we have an ester. And that's why your triacylglycerols are essentially considered to be triesters, because essentially you have 3 ester functional groups in the same compound. All right. So a little bit about some of the names of the triacylglycerols. So basically, with the fatty acids, you're sort of going to have to memorize them. They have a chart in the textbook, so you're going to have to sort of remember to what their structures look like. Basically, the glycerol part essentially becomes glyceryl. And if you think about it in terms of naming esters, right, we sort of use the branch name of the carbon group attached to the alcohol oxygen, which had a YL ending. So essentially glyceryl instead of glycerol. And then remember, for the carboxylic acid portion, we sort of changed the IC acid into ATE. So you'd have something like ethanoate or propanoate and such like that. Here, when the 3 fatty acids are the same, and I don't think you'd have to be able to name when it's a mixture, so I wouldn't worry about that so much. You have 3 steric acid molecules, so it becomes tristerate. And notice the ATE ending is similar to what we saw for the naming of esters themselves. I wouldn't worry about the common names, glycerol, tristerate, that would be essential enough. And again, I don't think you'd have to really know about naming the mixed ones. I think if you have ones where you have the same fatty acid 3 times and you understand the principles there, it should be perfectly fine. As we mentioned, your triacylglycerols may contain different fatty acids. So here you have steric acid, oleic acid, and palmitic acid. Honestly, the only way you would know that is by recognizing the structures of the fatty acids. This is an example of a mixed triacylglycerol. But it would be formed in the same way. You'd have 3 sort of esterification reactions using glycerol as the one alcohol portion. One of the things that's not as discussed as often is the fact that with these mixed triacylglycerols, you could really have isomerism. Sometimes when I look at a triacylglycerol, I sort of look at these as rows, sort of row 1, row 2, and row 3. But again, keep in mind, it's all one structure because the carbons of the glycerol are connected to each other. But sometimes when I view this, I do like to sort of look at this as sort of row 1, row 2, and row 3. And really, you could sort of change up the overall order, especially not for the ones where the 3 fatty acids are the same, but the ones that are essentially your mixed triacylglycerols. You could have different arrangements of connectivity. In my sense, really prevalent if you have 2 of the same and 1 different, because the 2 that are the same could sort of be in consecutive rows, or they could be sort of like row 1 and row 3, where the odd one is in the middle. So I think some of the older versions of the text, I know I'm using a different version than the one we use for the course, same types of concepts. But older versions of the text used to talk about that isomerism. Not so much, but just to make you a little bit aware of it. Triacylglycerols are the major form of energy storage for animals. Animals that hibernate eat large quantities of plants, seeds, and nuts that are high in calories. As the external temperature drops, the animal goes into hibernation. The body temperature drops to nearly freezing, reducing cellular activity, respiration, and heart rate. Not going to go any further than that. So, example here, again, just have to memorize the different structures. All right. Guide to drawing triacylglycerols. Again, esterification reaction. If we had this example here, take off the OH of each of the fatty acid portions, take off the hydrogens from the OH parts of glycerol, and bond each of those C double bond O carbons to the alcohol oxygen of glycerol. So when you put that together, you end up with your structure of your triacylglycerol. Okay. So we mentioned that your triacylglycerols are essentially your fats and your oils. Essentially, then what is the difference between a fat and an oil? Well, essentially, a fat is made up of many triacylglycerols. And an oil is actually made up of many triacylglycerols. But essentially, the key difference between them is that for a fat, the triacylglycerols that are found contain mostly saturated fatty acids. And that sort of leads to the property that they are solids at room temperature, right? Because if you think about saturated fatty acids, what do we know about saturated fatty acids? That they are essentially solids at room temperature. So if you, excuse me, if your fat is made up of many triacylglycerols containing mostly saturated fatty acids, it should make sense that they are usually solids at room temperature. So how does that compare to an oil? Well, as you might expect, an oil is made up of many triacylglycerols containing mostly unsaturated fatty acids. And again, that probably makes a little bit of sense because if you think about an oil and the description here of usually being a liquid at room temperature, then we know that unsaturated fatty acids, based on our discussion earlier, are usually liquids at room temperature. Now, there are some oils, such as palm oil and coconut oil, that are solids at room temperature. And basically what I usually say is that they have all the properties of being an oil, except that instead of being a liquid at room temperature, they're a solid at room temperature. And the main reason for that is if you take a look at coconut oil actually has a very high percentage of saturated fatty acids. But it's not usually classified as fat because essentially, I would say outside of the solid, even though it has a high percentage of saturated fatty acids, outside of sort of being a solid, it has more of the properties of being an oil rather than being a fat. Whether it's the source or just properties as well, it's considered an oil, even though it's a solid at room temperature. All right. So we talked about the saturated and unsaturated melting points. All right. So that's a little bit about that. Let's look at some of the reactions. Chemical properties. Okay. All right, so let's look at some of the reactions, some of the properties of triacylglycerols. So one of the reactions, and the nice part about the reactions that are here is that they are basically all reactions that we have seen previously. The first one that we're going to come across is hydrogenation. What hydrogenation essentially does is that it converts the carbon-to-carbon double bonds in the unsaturated fatty acids of triacylglycerols into carbon-to-carbon single bonds. Now, there's both complete hydrogenation and there's partial hydrogenation. And really, it's found very much commercially because you've seen on many product labels that this product, has been formed from being partially hydrogenated and such like that. So we can take oils like vegetable oil, hydrogenate them to give us stuff like margarine, in different types of forms where it's not a total solid, but it's a soft solid and such like that. So instead of having an oil within a liquid form, we have a soft or solid form. And health-wise, there's an advantage to this as well. So as we mentioned, in hydrogenation reactions, double bonds in the unsaturated fatty acids react with hydrogen gas to produce carbon-to-carbon single bonds. Just like we talked about with what? Just like we talked about with your alkenes. And I got to remember to start the pen. Just like essentially when your alkenes are converted into alkanes. The double bond between the carbons becomes a single bond. You add one hydrogen to one former double bond carbon, the other hydrogen to the other former double bond carbon, and that's it. Now, again, the process could be complete hydrogenation, where you break all of the carbon-to-carbon double bonds that are present in the triacylglycerol, or the triacylglycerols that are present. Or it could be partial hydrogenation, where you break some of the carbon-to-carbon double bonds. The way we add hydrogen is we bubble it into essentially the heated oil, because remember, your oils really are the ones that have the unsaturated, the carbon-to-carbon double bonds in the fatty acid portions, in the presence of a nickel or metal catalyst, which you need for hydrogenation. Sometimes it's tough to monitor the amount of hydrogen that's being added because you bubble it in, and it's a gas, so you can't see it. But there are tricks to make sure that the hydrogenation stops before all the carbon-to-carbon double bonds are broken. And that's what happens in commercial or partial hydrogenation. So what happens is that essentially not all, but some of the carbon-carbon double bonds are made into carbon-to-carbon single bonds. It changes essentially, if you think about liquid vegetable oil, the partial hydrogenation changes it to a soft semi-solid fat. The more saturated product has a higher melting point. I just want to see if it talks about the advantages of that. And I'm surprised. So one of the okay. So yeah, they do talk about the advantages of that. All right. Okay. So during hydrogenation, double bonds are converted to carbon-to-carbon single bonds. Now, a small number of the cis double bonds are converted to more stable trans double bonds, causing a change in the overall structure of the fatty acids. So this is a little bit different than what you would expect, because in essence, when the cis double bonds undergo hydrogenation, there's actually two things you can get. You could actually get single bonds between the carbons, or you can get trans double bonds. So essentially what happens there is an isomerization. So, a lot of times when it says sort of trans fat that's present, that's because in the hydrogenation process, yes, some of the carbon-to-carbon double bonds were converted to carbon-to-carbon single bonds, but in the same token, some of those double bonds which had the cis configuration, and essentially they all have the cis configuration, but some of them are converted into trans double bonds. You get an isomerization that occurs. All right. So in the body, trans fatty acids behave like saturated fatty acids. They're estimated to be 2 to 4% of our total calories. They raise the LDL cholesterol, but they lower the HDL cholesterol. So essentially, one of the things with partial hydrogenation is that, in a sense, it increases sort of the fat content that's present, because you're now dealing with saturated fat, but it's a little bit more heart healthier than products like that you obtain directly from animal sources. You think about butter, and I can't believe it's not butter. Why the heck do we want to use I can't believe it's not butter? Well, you see the heart healthy label on the bottle, because essentially that product, and you can get that in the semi-solid form, is made from partial hydrogenation. When you obtain butter, you obtain it directly from an animal source, which usually has a very high cholesterol associated with it. All right. So they show you the process here, hydrogenation, the addition of H2 giving the saturated product, and then of course the isomerization that converts the cis fatty acid into the trans fatty acid. All right, let's talk a little bit about hydrolysis. So where do we see hydrolysis previously? We saw hydrolysis with esters. And when we talked about esters, we saw both acid hydrolysis and we saw base hydrolysis. When we saw acid hydrolysis of an ester, what did we see? We saw the products were a carboxylic acid and an alcohol. Here, for a triacylglycerol, when it undergoes acid hydrolysis, the alcohol product is going to be glycerol. And instead of getting back one carboxylic acid, we get back 3 fatty acids. The way we would get the products would be exactly the same as we talked about with esters. You break the ester bonds, the glycerol oxygens pick up the hydrogens from water, and the C double bond O carbons pick up the OHs from water. And that leads you to the glycerol and the fatty acids. Base hydrolysis of a triacylglycerol is commonly referred to as saponification, because in a sense, what it is that it is soap making. Because what happens here? Well, just like with esters, when an ester underwent base hydrolysis, we talked about the fact that initially you get a carboxylic acid, but the carboxylic acid there wasn't in the final product because you can't have an acid under basic conditions. What happens to an acid under basic conditions? It becomes a carboxylic acid salt. Well, the same thing here. Initially, what we do get back, is we get back, instead of 1 carboxylic acid, we get back 3 fatty acids. But again, you can't have these fatty acids under basic conditions. So what happens to the fatty acids? They become fatty acid salts, as the oxygens from glycerol essentially pick up those hydrogens of the 3 fatty acid molecules. Those 3 fatty acid salts are commonly known as soap. If you use sodium hydroxide, you get a solid soap. Use potassium hydroxide, you get a softer liquid soap. All right? So in this case here, just to show you, when this would break, you would get 3 of the same fatty acids. But again, because we're under basic conditions, we can't have a fatty acid as the product. We know for a carboxylic acid and now for a fatty acid, which part is the acid part? The acidic part, it's the hydrogen of the carboxyl group. That's the acidic part. And essentially, those hydrogens are going to be picked up by the glycerol oxygens, because essentially these OHs here make up the OHs on the fatty acid portion. So that's why you see the darkened one here and the darker one here, because essentially these take the place of the glycerol portions. So you get 3 fatty acids. And then when these hydrogens come off, instead of having C double bond O connected to OH, instead you have C double bond O connected to O minus. And what else is going to be present, attracted to those O minus charges, are going to be the sodium ions that are also present. And that gives us our soaps. All right. So we got some of the different reactions that are here. So definitely want to be aware of these reactions. Absolutely. All right. So let's jump to, let's go to phospholipids. All right. So phospholipids are similar to triacylglycerols, and there are basically 2 main types. There are glycerol phospholipids, which we'll start out with first, and then there are sphingomyelin. For a glycerol phospholipid, the backbone of the structure is still glycerol. And connected to the glycerol at 2 portions, you have fatty acids. But the difference is, for a glycerol phospholipid, in the 3rd portion or the 3rd row, you don't have a fatty acid. Instead, the glycerol is connected to a phosphate group, and the phosphate group is linked to an amino alcohol. That's the general structure of a glycerophospholipid. Okay. So what we'll see is that the glycerol, the phosphate, and the amino alcohol are connected to each other by what is known as a phosphodiester bond. And it's going to be good to become acquainted with that to a certain extent, because when we get to the chapter on nucleic acids and we see how your nucleotides are formed when nucleic acids are linked together, they are linked together by phosphodiester bonds. Okay. So again, the glycerol and the fatty acids are linked together by ester bonds, but the glycerol, the phosphate, and the amino alcohol, they're linked together by phosphodiester, or they're linked to each other by a phosphodiester bond. In comparison to a sphingomyelin, for a sphingomyelin, the backbone is not glycerol. Instead, the backbone is a structure called sphingosine. The first part of sphingosine, which is like this part in blue up here, doesn't react with anything. It sort of stays the same. As we'll see, one part here reacts with the fatty acid, not to give an ester functional group, but to give an amide functional group. So the sphingosine connects to a fatty acid in the middle here by forming an amide bond. And on the bottom, the sphingosine is going to connect to phosphate, which is going to connect to amino alcohol, and that's going to be through a phosphodiester bond. So just like with a glycerophospholipid, down here on the bottom, between the sphingosine, the phosphate, and the amino alcohol, you're going to get a phosphodiester bond. All right. So let's go back to glycerophospholipids. The way that you essentially recognize the different types of glycerophospholipids is based on the amino alcohols that are present. And there's three possible amino alcohols that are present. There's choline, there's serine, and there's ethanolamine. And the reason why they're classified as amino alcohols, when we take choline, is that you have an alcohol portion and you have an amine portion. Now, as we'll see, serine is not only a amino alcohol, as you'll see in the next chapter, it's also an amino acid. Now, based on our body pH, which is 7.4, these species are actually not in a neutral form in the body. They are in an ionized form. And that part really has an important significance that we'll touch upon, I'm sure, shortly. So these are the three different amino alcohols that are present. And really the way we distinguish between glycerophospholipids is based on what amino alcohol is present. Okay. So the two main types of glycerophospholipids that you come across are what are known as lecithins and what are known as cephalins. The way we identify a lecithin is that the amino alcohol that's present in the structure is choline. And the main way that we identify a cephalin is that the main amino alcohol that's present is ethanolamine. Sometimes the cephalin can contain serine, but most of the time the cephalin will contain ethanolamine as the amino alcohol component. So if you look at the two structures, you've got your fatty acid portions. Now, where's the phosphodiester bond? So here it is. Okay. P double bond O, OC once, OC twice. How do we come up with that? If you think about an ester, C double bond O connected to OC. We have what are known as phosphate esters. For a phosphate ester, instead of having C double bond O, OC, we have P double bond O, OC. Now, for a phosphodiester bond, that P double bond O group is involved in two ester linkages simultaneously. So P double bond O, OC once, P double bond O, OC twice. So that essentially is your phosphodiester bond. Okay. So One of the big things about lipids in general is that for the most part, when you think about lipids, lipids are thought of as being nonpolar species. Waxes and triacylglycerols, fats and oils, nonpolar. But glycerophospholipids contain both a polar region and a nonpolar region, which allows them to interact with both polar and nonpolar substances. Okay. So, one part of the glycerophospholipid is what is known as the head of a glycerophospholipid. Okay. And a lot of times glycerophospholipids are sort of drawn in this sort of crazy way to show the two different regions, what is known as the head and what is known as the tail, sort of indicated by this diagram on the bottom. The head of a glycerophospholipid is polar because you have atoms that have charges associated with it. We talked about how in the body, the amino alcohol is going to exist in an ionized form. Well, also the phosphoric acid in the body doesn't really exist as H3PO4. It actually exists as its conjugate base, H2PO4 minus, which is this structure right here. And if you look at that structure, one of the oxygens has a negative charge. So when you link the choline, the phosphoric acid, and the glycerol together to give the phosphodiester bond and the head of a glycerophospholipid, you have two atoms that have charges. So this is a polar region. What substance in which most lipids won't be soluble in is this region going to be soluble in? Water, right? Based on the idea of like dissolves like. You have atoms that have charges creating a polar region. Water is a polar substance, so this part can dissolve in water. Then the fatty acid portions make up what is known as the tails of a glycerophospholipid. And as we know, carbon chains themselves are nonpolar. So the tails of a glycerophospholipid is essentially the nonpolar region. So now this part is not going to be able to dissolve in water. However, other nonpolar substances, like what? Like other lipids, can dissolve in this region based on the idea of like dissolves like. And as you saw in the chapter, what do glycerophospholipids really help in? They help in lipid transport because, as you know, our bodies are mostly composed of water, and you have a polar region which is going to dissolve in water, and you have a nonpolar region which is not going to dissolve in water, but can dissolve other nonpolar substances. And essentially, when the head dives into the water, the tails come along for the ride. And so you have transport that takes place. All right, so a little bit more about sphingomyelin. As we talked about, the backbone is a structure known as sphingosine, which is a long chain amino alcohol that replaces glycerol. As we mentioned, you get an amide bond between the amine group on sphingosine to a fatty acid. Remember, as we saw, how do we usually get an amide? Carboxylic acid plus amine or ammonia or primary or secondary amine. And then we talked about the phosphodiester bond. Now, the two amino alcohols that you generally see in a sphingomyelin are choline and ethanolamine, not so much serine. All right. Reason why they're important: sphingomyelins are abundant in the white matter of the myelin sheath, the coating surrounding the nerve cells that increases the speed of nerve impulses and insulates and protects the nerve cells. In multiple sclerosis, sphingomyelin is lost from the myelin sheath, which protects the neurons in the brain and the spinal cord. The myelin sheath deteriorates as the disease progresses, and scars form on the neurons and impair the transmission of nerve signals. So, multiple sclerosis, very serious. So it involves actually losing sphingomyelin from the myelin sheath. So that's sort of the importance of that. Show you a structure here of a sphingomyelin. Okay, so in orange is the structure of essentially sphingosine, not the total structure. Sorry. If it was sphingosine, there'd be another hydrogen on the nitrogen, and there'd be a hydrogen on the oxygen. Notice here in the middle, C double bond O connected to nitrogen. What functionality is that? That, of course, is an amide. And then on the bottom, you have right here, P double bond O, OC once, OC twice. What functionality is that? That is your phosphodiester bond. All right. Let's go a little bit into the last type of lipid that we come across, which are your steroids. Okay. So your steroids essentially are the one type of lipid that are not essentially esters. Really, it's very difficult to miss a steroid in a sense because of the steroid nucleus. What you have are 3 cyclohexane rings and 1 cyclopentane ring that are fused together. What does it mean to be a fused ring system? It means that you have overlapping sides, or you have rings that share a common side. So here, I did it again. Here, here, and here, you have 2 rings, or you have rings that have a common side. That's when you have a fused ring system. All right? And really, to distinguish the rings, I wouldn't really so much worry about the numbering aspect. The main way to distinguish the rings is as A, B, C, and D. Those are what is known as the A ring, the B ring, the C ring, and the D ring. All right. The steroid nucleus, usually the methyl groups are, I usually haven't seen the methyl groups too much there. I mean, in specific lipids, they could be there, and I think they are there in certain cases. But really, they use that, I guess, to indicate the numbering system. But I'm really used to just using A, B, C, and D to distinguish the rings, especially between the 3 cyclohexane rings and even with the cyclopentane ring itself. Most important, well, one of the most important steroids, cholesterol. All right. The biggest thing about cholesterol is that it's classified as a sterol. And the reason why it's classified as a sterol is because of the OH group that's attached to the A ring. I know it says carbon 3, but it's attached to the A ring. And essentially, the main form of cholesterol in the body is in the form of what are known as, I'm surprised they don't mention it. So the main form of cholesterol in the body are what are known as cholesterol esters. And essentially, how do you get your cholesterol esters? Well, cholesterol undergoes esterification with fatty acids that are generated in the body when your fatty acids undergo hydrolysis. When your triglycerols, undergo hydrolysis. Because in the body, essentially, they're going to undergo acid hydrolysis, but instead of sort of using acid, enzymes like lipases are going to break them down instead. So obtained from these, synthesized from the liver. Diet that is low, recommends that cholesterol seeds foods, cholesterol content of some foods, solution. All right. So yeah, so cholesterol really is the main type of one of the most important lipids. Keep in mind, one thing of importance, synthesized in the liver, okay? Sometimes you're asked about where it's made from. So you want to remember that cholesterol is synthesized in the liver. That's an important point to remember. Bile salts. So bile salts are another type of steroid. So they are also synthesized in the liver. The liver plays a key role, all right? And they're actually made from cholesterol, and they're stored in the gallbladder. Now, bile salts actually have polar and nonpolar regions. So they can also interact with polar and nonpolar substances. Essentially, to me, your nonpolar region is really the steroid nucleus. Even though there's OH groups that are present, you know, the number of carbons that are in the steroid nucleus really sort of make it a nonpolar region. But then the reason why you have it classified as a salt is because on the end you have a you have a carboxylate group. the C double bond O connected to O minus. And then a lot of times in these bile salts, you'll really have some sort of counter ion, a cation, to be attracted to that O minus charge. And that part is ionic and that part is polar. So your bile salts are able to interact with both polar and non-polar substances. But the steroid nucleus is here, and that's exactly why it's classified as a steroid. All right. Lipid transport. So as we mentioned, most lipids are non-polar. And of course, here they show you the cholesterol ester, which they would have showed a little bit earlier, but okay. So the way that, in a sense, lipids are made more soluble is by combining them with glycerophospholipids and proteins to make water-soluble complexes called lipoproteins. And really, it's in the form of these lipoproteins that lipids are transported through the body. Lipoproteins are spherical particles. You have a polar surface, and then you have an inner core. Here's a blow-up of the polar surface. What makes the surface polar? Well, essentially, you have the heads of your glycerophospholipids. Okay. You also have essentially the OH portions of cholesterol, right, on the A ring. Even though there's large rings that are present, that OH group would sort of want to migrate to the surface to be close to water. Really, to me, it's the heads of the glycerophospholipids are, and essentially and even the sphingomyelin in a sense. I don't know, we don't really talk about so much as sphingomyelin as having a head and a tail, but you really do have polar and non-polar regions of a sphingomyelin as well. And then also protein. Protein in a sense tends to be polar. The outer surface of the lipoprotein, is going to dissolve in water. Now, what do you have inside in the inner core? Essentially, you can see by the drawing here, you have sort of the carbon portions of the steroid, the cholesterol, the rings of the steroid nucleus, and you have the tails of the glycerophospholipids. What types of things are going to dissolve in those tails? Well, most of the other main types of lipids, like your cholesterol esters and your triacylglycerols. And really what that outer surface does is that it not only helps to move the lipids through the body, which is mostly aqueous, but it protects them from water on the surface as well. So the outer surface is able to dissolve in water, and when it moves through the body, the lipids that are on the inside, the non-polar ones, come along for the ride. So it's really in the form of these spherical particles known as lipoproteins that lipids are transported through the body. All right. Different types of lipids you're probably familiar with, LDLs and HDLs. We have them monitored. You also have very low density very low density lipoproteins, which are your VLDLs. Then you also have your chylomicrons. The way that lipids differ is in their density, composition, and function. All right. So lipoproteins such as HDLs and LDLs transport non-polar lipids and cholesterol to the cells and liver. HDLs are the good cholesterol. The LDLs are your bad cholesterol. So we usually have those checked, and on the slide here, they give you some numbers, recommended levels, and greater risk of heart disease and such. All right. Steroid hormones. So testosterone, progesterone, really what they are, they're all steroids. They're produced from cholesterol. They're essentially chemical messengers that serve as a communication system for the body. You have the male sex hormones, testosterone and androsterone. You have the female sex hormones, estrogens and progesterone. You also have what are known as adrenal corticosteroids, which are produced from adrenal glands, mineral corticosteroids, glucocorticosteroids, and such like that. Show you some examples: testosterone, estradiol, progesterone, norethindrone. They're all steroids. They all contain the steroid nucleus. The difference between them, is the different functionalities that are present. Here you sort of have ketone, alcohol. Here you got phenol, alcohol. Here you've got a couple ketones. Here you got an alkyne with an alcohol and a ketone. So in a lot of the different steroid hormones, it's the functional groups that are present in addition to the steroid nucleus that really identify them from each other. Anabolic steroids, used a derivative of testosterone, used illegally to increase muscle mass. So unfortunately, a lot of people, some people at the gym tend to take these. And just to show you, they're also steroids. You can see the steroid nucleus that are present in these structures as well. Adrenal corticosteroids produced by the adrenal glands located on top of each kidney includes aldosterone, which regulates electrolytes and water balance by the kidneys. Also include cortisone. You might have gotten a cortisone shot at one time, which increases the level, increases blood glucose level, and stimulates the synthesis of glycogen in the liver. All right, and those are the main types of lipids. All right. And I think that's what we have time for today. All right. There is one more section on the cell membrane. I'll see if I can get a little bit of time to post a little bit of information about that. I don't want to ignore that aspect of it, but for now, those are really the main types of lipids that you come across. All right. Any other questions? All right. Stay well and hope to talk to everybody soon. Have a good night.